you know where they want to go? But they're all polar, aren't they? They all need a channel to move through. No channel, you're screwed. You're not going to move. You're not going to get in or out of our cell. So for us, we're going to say that cells are more negative on the inside than anything else. If you add up all the positive charged things inside of a cell, and you have all the negative things inside of a cell, there are more negative things than there are positive things. And one of the biggest contributors to that negative charge are albumins. They tend to have a negative charge to them. These little protein structures that are inside of a cell. And overall, we're going to make our cell very negative. And we're going to show it like this. I'm just going to give you a number for the moment of negative 70 inside. Negative 70 millivolts. Now we have this term up here, right, called the membrane potential, right, measured in millivolts. That is going to be the difference in charge across the cell membrane. The difference in charge across the cell membrane. And these charges line up right on the underside. They're trying to get to the positive charge thing is on the outside. So we kind of think of a cell membrane like a battery. And since there are more negative things, they're trying to get to the positive things. And the positive things are trying to get to the negative things, but they're polar, and cell membranes don't let polar things cross. So this creates a charge across the membrane. We call it the membrane potential. And let's draw another one right here just for a moment. Let me do this. And let's say in cell A, it's negative 70. And if I draw this, make it a bit bigger. What's going to happen to the charge inside of cell B because potassium is leaving? Become more negative, right? Because we're losing positive charge. Everybody okay with that? All right, awesome. All right, so maybe it'll become a negative 72. What if we did this? Minus 70. We'll get to the chase right here. What happens to that number if sodium comes in?
Becomes more positive.
It goes up. Becomes negative 71.
69.
So it becomes, and here's the way, two ways to say this. It becomes either less negative or more positive. It says the same thing, right? Either more positive or less negative. So it could become maybe minus 68, right? That's less negative. That's also more positive. And it was simply due to ions crossing the cell membrane. Hello, guy.
I bother you for
Yeah, give it a
Can
second.
I borrow him for 30 seconds?
He's my boss.
The food's here. Amy has it, but I want it. Look at him just being out here. Walking, holding me back McDonald's. Don't tempt me right now. You're cute. Keys.
So, that's the beam.
What?
Yeah.
Thanks, boss.
All right. Saddle City Light is shutting power off to the campus next Monday at 5 p.m. for the whole frickin' night. Yeah.
They suck.
To do some wiring. So, he's going to send me an email to remind you guys, but he wants wanted to let me know now, so I can let you guys know right now. I'll figure out what I'm going to do, because I want to be fair to everybody. So let me worry about this, but for right now, the power's going to be shut off Monday from 5 o'clock on. What I think I'm going to do, that's something I'm doing, is going to do a video lecture and post it for you guys on Monday. So that means then is, just to be fair to everybody, for those of you that can and want, I'll come here for the first lab section, but I won't do any lecturing. So in other words, Monday from 3:15 to 4:40 or 4:59 is open lab. All right, come if you want, don't come if you don't want to, right? And then I'll try and get a couple of video lectures up for you guys so you don't fall behind on our schedule. But basically, Monday, no school. Okay? All right. So back to this, right? So sodium coming in makes it become less negative, more positive with that. So what causes the membrane potential to change? The movement of ions across the membrane. All you need to know is what ion was moving and in what direction. That's all you need to know. That'll tell you how the membrane potential changes. All right, so we keep these things in mind. I know what hydrophobic is. I know what hydrophilic is. I know membranes are semi-permeable structures. I know that transporters are channels. They can be either open, in which case there's going to be a nice little balance between all of this. Now, one thing that they're not showing you, that I want to add one more picture to drum this into you, is this. So here is going to be what's going on inside of any cell, neuron or skeletal muscle cell. Give me a moment to get this drawn before you copy it down. One last thing. I'll get out of the way and read this to you. All right, this blue circle is called a sodium-potassium ATPase pump. Memorize that name. And memorize this, in case you weren't taught this in 211. It's going to take 3 sodiums at a time, and it's going to pump them straight out against the concentration. That means it's an active transporter. Once it's done pumping the 3 sodiums out, it then grabs 2 potassiums, and it pumps them back in, again, against the concentration. It's like a carnival ride. 3 sodiums out, 2 potassiums in. It costs you 1 ATP to do this. Right? It is an active transporter. Now, why do we need pumps? We need pumps to maintain the concentration gradient of whatever ion we want. Or I should say, whatever ion it moves. Right? If I have an equilibrium potassium, potassium won't diffuse out. I can't change the membrane potential. Equilibrium of sodium. Sodium won't diffuse in. I can't do a membrane potential. So this pump keeps pumping sodiums out, so sodium can always diffuse in. It keeps pumping potassium in, so it will always be able to diffuse out. And this is going to be our neurophysiology. This right here is going to make muscles work by letting potassium go out and let sodium come in.
Is there potential before the movement or after?
For what kind of movement?
The membrane potential, is that after the ions have moved or?
That's during the ions moving. So every ion that moves causes a change in the membrane potential. It's constantly changing, right? And we can change it a lot or we can change it a little. So here's what I want you to think about. That kind of gets to your question right here. This is what's happening in your cells right now. Remember, there's no doors to these. They're following the rule of chemistry. Potassium is always diffusing out, and sodium is always diffusing in. But it's being balanced with the pump, pumping 3 sodiums out and 2 potassiums in. And think of this as your engine idling at a stop sign, right? It's just idling. And we're going to stay at a certain resting value, right? This is our resting membrane potential. We're just letting it exist all by itself until we screw it up intentionally. But if you just leave the cell alone, like right now if I can stand here and my biceps aren't working, right? There's a resting membrane potential of minus 90 millivolts across those cell membranes. When I just did this, I rapidly changed it. And we'll talk about how we do that in a couple of minutes from now. But if you keep this in mind, right, that a resting membrane potential is due to the constant diffusion of potassium out along with sodium in, along with, it's a dance, right, with the pump running. That given that certain ratio, I'm going to stay at minus 90 for our muscle cells. So our muscles are actually at this value. But if I screw around with this, I screw around with that. And that's going to be the goal.
Isn't that possibly 1 ATP?
Yeah, that 3 sodium to potassium. That's 1.
So the pump running is what's allowing it to come in and out.
Well, yes and no. It's maintaining the ability. What's allowing it is the concentration gradient.
Okay, so
The difference in amounts is what's actually allowing it. But I'm maintaining the allowance with the pump.
the pump is like, I'm gonna put some out, and then that channel up top is like, okay, well, I'm gonna put some in then.
Right.
And then that one goes, I'm gonna put some in, okay, I'm gonna put
You ever
some
seen a
out
ship
now.
at the, like a cruise ship on the piers? There's always water being pumped out of the hull, right? It's a pump, so because there's water always leaking in.
Yeah.
If the pump didn't run, what happens to the ship eventually? It sinks,
Yeah.
right? So my pumps are always running. Okay. Let's see if we can put this into action then. All right. Here is a neuromuscular junction from Monday night. Let's get our bearings with this. So there's the motor neuron, right? This is the end of the motor neuron. That gold line you see is the cell membrane. So what pops in your head? This. The idea of a semi-permeable membrane pops into your head. The idea of concentrations of ions trying to cross the border. Sodium trying to come in, potassium trying to come out, calcium trying to come in. And inside you have these little blue things that are called what? Acetylcholine vesicles, right? They're a transport vesicle. Remember what a transport vesicle is comprised of? Yeah, a phospholipid barrier structure, right? It is a little balloon of phospholipids that contain a neurotransmitter, a chemical. I need to get that little balloon to release its chemical substance. So how do we do this? Here comes the, just trust me on this statement. Because we have, this is like talking about a circle, where a circle begins. A wave of electricity is passing along the membrane, right? And that wave of electricity is called an action potential. It is simply just a really quick change in the membrane potential. And we'll get to understand it by the end of tonight. So a wave of electricity, a wave of voltage travels along the membrane. And think of it as a series of dominoes. That's the best way to think about it. If my red light is the wave of electricity traveling along the membrane, then this table is like the membrane, and I'm traveling this way, knocking down dominoes one after another, traveling along the length of the membrane. And as this voltage travels along the membrane, it then comes across and hits a voltage-gated channel. Well, what is this voltage traveling on the membrane going to make that voltage-gated channel do? Open. All right. Now, what's going to happen because it's open? Well, now calcium is going to go through it because you opened the door. And here's what you got to think about, the way you start thinking about physiology. I know that the voltage-gated channel is opened by voltage. That's why they called it a voltage-gated channel. I know it's for calcium because they named the ion for what the channel will allow to pass through it. So it's a calcium voltage-gated channel. Nobody else can get through it. This is a members-only club idea. So once I open up a calcium voltage-gated channel, and John would say, well, why is calcium crossing the membrane? It's not because the gate's open. It's because of why?
Diffusion.
It's following its diffusion pathway. Because I can open up the gate to have an equilibrium created, and calcium won't move, right? Ions only move because of a gradient. Ions only move because they're following their constant, or they're going down their concentration gradient. That's the only reason they move. We just allow them to do it by opening up the voltage gated channel. So here comes this electricity traveling along the membrane, right? It hits a voltage gated channel, there's lots of them, right? And then that allows calcium to come in. Well, what does that calcium do? Well, here's calcium, and calcium does this. PowerPoint's so fun. Now, look where I made calcium stop. It stops on an acetylcholine vesicle. It attaches to it. It must do that. Now that calcium is attached, you can then do, does anybody know what the term is called for this process right here? That's exocytosis, right? Calcium must come in. It's a calcium-dependent process. You do exocytosis. No calcium on the vesicles, no exocytosis. So now, you keep in mind this, right? This is the fluid outside of a cell. That means there's tons of sodium out here, right? There's my muscle's membrane. That means there's tons of potassium in here, but not a whole lot out there. Now, I want sodium to come in. So how do I get sodium to come in? Well, there's sodium. If I look carefully at these diagrams, right here, I realize that that bluish structure has a notch right there, but there's nothing in the notch. That one has a notch, nothing in it. That one has something in the notch. That little blue ball or purple ball is one of those molecules of acetylcholine. That's a chemical. So since that landed on this and made it open, what kind of a transporter is this? It's a chemical gated, right? It's intuitive now. Right? And they're only showing you one because that's the only one that has a molecule of acetylcholine attached to it. And now I understand why sodium is coming in, because there's way more sodium outside than there is inside. It's simply just obeying the rules of diffusion. But now, what happens when sodium comes into the cell? What changes? The membrane potential becomes more positive. The more sodium, the more positive it becomes. Now, the truth of the situation is, where acetylcholine, there's a couple of ways to talk about this. That is a chemical gated sodium channel. It's also known as an acetylcholine receptor. It's the same thing, but acetylcholine receptors are chemical gated sodium channels that only open when acetylcholine attaches to them. Right? So it's an acetylcholine channel or a chemical gated sodium channel. Now also look at the picture here. What do you notice about the sarcolemma, about the muscle's membrane? What do you notice about it? What did the artist do to it?
It's a green and blue.
Two different colors? Why the hell they do that? Because look where it's turquoise, right? This part right here. That's what's known as the motor end plate. It's a part of the muscle's membrane directly beneath this motor neuron. And in the turquoise part, you only have chemical gated sodium channels. Bless you. There are no voltage gated channels there. You only have chemical gated channels. Now, where I'm pointing at with my laser pointer here, are there any sodium leak channels in there? What did I say about 2 minutes ago? There are what? There are always leaky channels for sodium, potassium, even when the artist doesn't show them to you. Right? You burn this into your head. There is always sodium, potassium leak channels. There is always sodium, potassium leak channels. There's always sodium, potassium, lead channels. Then why in the hell did the artists put them in? Because most artists only put in what the focus of the conversation is about. Because it gets too muddled to put everything in there, right? And I can show you the cytoskeleton in there as well. I'm not, right? I can show you cholesterol in the member. I'm not. It gets in the way of the conversation. So you got to remember that there are certain things that are always implied but not shown. So there's always going to be sodium, potassium, lead channels. Are there going to be any of these things in the membrane?
Yes.
Oh yeah, hell of a lot of them. But they're not showing them to you. But they're just implied. You have to know that they're always going to be in the membrane structure. So you just burn this into your mind. All membranes have this for sure, unless otherwise told. All right, now, the more bluish part, right, out from under the motor neuron, right, this part here and that part there. Look at the actual structure and also look at the name. You see that little doorknob right there? See how there's not a doorknob over here? Because these are chemical gated and these are voltage gated. You only have voltage gated channels out here. No chemical gated. No chemical gated with the blue and no voltage gated with the turquoise. Now, also in the turquoise part is an enzyme known as acetylcholinesterase that helps to break apart the neurotransmitter called acetylcholine. It's like an off switch. It helps me to stop acetylcholine from landing on these chemical gated channels. It allows me to kind of control how many of these channels actually get opened up. All right, go ahead.
Sorry, does that enzyme bind to the receptor the same as the acetylcholine to change the...
No, it actually binds the acetylcholine
Okay.
and breaks it into its two substrates, acetate and choline, which we don't have to worry about the actual name. All right, so let's back up. Go to the beginning again. This thing called an action potential, right, is a wave of voltage traveling along the membrane of that motor neuron. When that wave of electricity, called an action potential, comes across any voltage-gated channels, like a jack-in-the-box, it pops them open. And then whatever channel that ion is for, ions will do whatever they need to do. Since that's a calcium channel, then calcium floods into the motor neuron. As calcium comes in, it attaches to these vesicles that contain acetylcholine. Because calcium is attached, right, that allows the vesicle to fuse with the membrane and do exocytosis, raining neurotransmitters onto the surface of a motor end plate. So I've got all these neurotransmitters now raining down here. When, by pure frickin' chance and luck, a neurotransmitter lands on a chemical gated channel, it makes it open up. Because when chemicals bind to proteins, they change their shape, right? So I changed the shape, I'm now open. Why does sodium go in? Not because you've opened the door, because it's following its concentration gradient into the cell. And because sodium has a positive charge, I make the membrane less positive. I'm sorry, less negative, more positive. And the more sodium comes in, right, the more positive I make it. Now, there's other idea we got to talk about. Let's make this our cell membrane, and this is a chemical gated sodium channel that's opened up, and I'm going to be a puff of sodium ions. Not a single, but like a little cloud of sodium. Because somewhere like around maybe 15 to 25,000 atoms cross really quickly through that one little channel, like little mini Niagara Falls. So big pile of sodium is right here. And let's freeze time for a moment. Right, there's a big pile of sodium right here. Where am I going to go? Every frickin' where there's less of me, right? I'm going to go that way, I'm going to go that way, I'm going to go that way. Wherever there's less, I diffuse to where there's less of me. Right? But as I diffuse to where there's less of me, there actually becomes less of me. Right? My amount gets smaller and smaller and smaller, which means my effect gets less and less and less. It's sort of like this idea. I don't know if we're going to be able to do this. Let's see if we can. Okay. Can you hear me, Tess?
Yeah.
I was talking about you, Michelle.
Yeah, I heard Michelle, but I was just like, I can't hear a word you're
Okay,
saying.
exactly, right? Why? What happened to the strength of my voice the further it got from my mouth? It got weaker and weaker, right? That's what's happening to the diffusion of ions, right? Imagine a little cloud, a little concentration of them, but as they spread, their effect gets less because they're further from their point of origin. That's what's happening here. Sodium can diffuse in both directions. It's going to go that way in the diagram, it's going to go this way in the diagram. But as my cloud of sodium diffuses, hello, hey, thank you. Come on, thank you, keep going, there you go. All right, I finally diffused way over here, and if there's enough of me to raise that membrane potential from -90 to maybe -55, I open up the voltage-gated sodium channels, right? -55 is the magic number. It's called threshold. If I don't reach threshold, it's not going to open up. So -55 is threshold. It's the same idea that if you guys have air conditioning in your homes, right? At what temperature does the air conditioner come on? Maybe it's set to 80 degrees, right? So if it gets to 81, the AC turns on because the temperature reached the threshold to turn on the AC. The membrane potential reached a certain voltage, which happens to be -55, right? And that made it open up those voltage-gated sodium channels. Remember, we went from -90 to -80 to -70 to -60, -59, 58, 57, 56. We haven't opened the door. At -55, that door pops open. I don't care if you screen pass, it gets to -10. You'll open up. As long as you get to 55 and more positive, those doors open up. And when those doors open up, you get a lot of sodium coming in. Look how much bigger my sodium symbol is compared to this one, right? I'm talking Niagara Falls, Amazon flood amount of sodium. So you get a big change in membrane potential, up to about a +30. So some of the ways to keep this in mind, help you understand this process, some basic rules. Ions can't cross a membrane, over here on the whiteboard, unless they have a transporter in place for them. If we're talking about gated channels, then the gate has to be opened up. Whenever ions do cross a cell membrane, think about the charge on the ion and which way the ion is diffusing. And you can figure out the actual effect on the membrane potential. Keep in mind that chemical gates only open because of chemical, and voltages only open because of voltage. What we have here is a relay system. I opened up a bunch of chemical gated channels, let some sodium come in, and that diffused out from under the motor end plate until I finally got to the part of this arc lemma where there's lots of voltage gated sodium channels, and there was enough of me to actually reach threshold and open up a batch of voltage gated channels. So I need both for potassium and for sodium in a moment, as soon as I get to Brian's question.
All of the chemical gated channels specific to a single ion. So like sodium gated channels just do sodium head. Or are there some channels that do multiple ones?
Yeah, see, this one lets a bit of potassium out at the same time.
Okay, so they can.
It depends, all right? In this scenario, these guys let a little bit of potassium through, but the majority is way more sodiums come in than potassiums leave.
So you call it a sodium gated channel.
Yes, because
And
it's mainly
primarily
allowing
sodium.
for the net diffusion of sodium in with a pinch of potassium out.
Is there a potassium gated channel?
Not for chemicals, no. Not in the motor end plate. Not in this turquoise region. There's not.
Okay.
Okay. Is making sense so far?
Yes.
Okay. Let's do this. 5-minute break. Take 3 minutes of that break. breaking